Hi everyone, so in this video I'll talk about application of genetic engineering. So let's get started. So we'll talk about the first application of genetic engineering, which is the production of recombinant pharmaceuticals. So talking about a small intro about what it is all about and how it is helpful in the application of genetic engineering. So the number of human disorders can be traced to the absence or malfunction of a protein normally synthesized in the body. And most of these disorders can be treated by supplying the patient with correct version of the protein, but for this to be possible, the relevant protein must be available in relatively large amounts. So if the defect can be corrected only by administrating the human protein, then obtaining the sufficient quantities will be a major problem unless donated blood can be used as a source. So in this case, we'll majorly talk about insulin or formation of recombinant insulin as the first application for genetic engineering. So this is a major application which is the production of recombinant pharmaceuticals. So first we'll be talking about insulin and how it helps in forming recombinant insulin and solving the problem of supplying insulin to the diabetic patients. And we'll also tell you about how recombinant insulin is far more better than the normal insulin that we have. So talking about the animal proteins are therefore used whenever these are effective, but these are not many disorders that can be treated with animal proteins, and there is always the possibility of side effects such as allergic response. So we can always use animal products so as to treat ourselves, but there are contaminants that may affect us due to presence of system. There are something or some parts which are lacking in humans and animals, and there are differences between humans and animals and their products as well, so as to we cannot use them anywhere and everywhere. Also, gene cloning can be used to obtain large amounts of recombinant human proteins. So gene cloning is the only technique or the most useful technique used in the genetic engineering for maintaining a balance for maintaining a pure amount of proportion of all the pharmaceuticals that is being produced in a recombinant manner. So moving with that, so talking about the recombinant insulin that we have here. So insulin, which is synthesized by the beta cells of islets of Langerhans in the pancreas, controls the levels of glucose in the blood. Also, an insulin deficiency manifests itself as diabetes mellitus, also a complex of symptoms which may lead to death if it is untreated. So insulin deficiency is observed in diabetic patients and which may lead to diabetes. So we need to sort this problem, right? So people are generally given insulin who are diabetic. So there is a concept of recombinant insulin that is brought in with the help of genetic engineering, so that we can sort this problem even in a better manner. So fortunately, many forms of diabetes can be alleviated by continuing programs of insulin injections and thereby supplementing the limited amount of hormones synthesized by the patient's pancreas. So definitely insulin is not available in abundance, so not always do we get insulin. So the concept of recombinant insulin has been brought in so that it may contain that many enzymes or hormones so that the human pancreas or the human gut can synthesize. So the animal's insulin is generally satisfactory, but problems may arise in its use to treat human diabetes. So the one problem that arises due to use of animal insulin is the slight differences between the animal and the human proteins, which can lead to side effects in some patients. And secondly, we have the potentially dangerous contaminants which cannot be always be completely removed during the filtration process. So these are major threats to human life. So that is why animal insulin is not always preferred over normal insulin. Also, the insulin used in this treatment was obtained originally from the pancreas of pigs and cows slaughtered for meat production. So initially, to solve the problem of insulin, or the cause insulin is not available in abundance and it has a limited supply, so to sort this problem, initially the scientists used the animal insulin, so which was obtained from the pancreas of pigs and cows, but which was not a very successful method, you can say. So thereby we came to the concept of recombinant insulin. So how can insulin be used as a as in a recombinant mode, or we can use in a gene cloning manner to produce more and more recombinant insulin? So the two features of insulin that facilitates its production by recombinant DNA techniques. So the insulin by in itself is very versatile, so that it can be used for production by recombinant means. So some of the features that it contains, and which is very useful for us to use it as a recombinant source, which is the first is that the insulin is not modified after translation by the addition of sugar molecules. So the recombinant insulin synthesized by the bacterium should therefore be active. So after translation there is addition of no sugar molecules additionally to the normal insulin molecule. Alright, and secondly is the size of the molecule. So the insulin is relatively small protein comprising of two polypeptides only, one of 21 amino acids and secondly of a 30 amino acids. So you can see this insulin molecule. All right, so one is the 21 amino acid, so you can also name it as A chain, and we have the second chain, which is the B chain, which consists of 30 amino acids, and both of them are linked by two disulfide bonds or linkages. You can see disulfide bonds which are linked together with the A and the B chain. So in humans, these chains are synthesized as precursor called pre-proinsulin, which contains A and B segments linked by a third chain. So I'll explain you how humans synthesize these type of insulin. So how humans get the insulin, right? So these form of insulin are available in this form. This is a huge chain, you can see. So it consists of both the chains A and B. So this is the A chain, this is the B chain. It also consists of another chain, which is the C chain. Alright, so it consists of a C chain, and it also has a leader sequence. Alright, so it starts with a leader sequence, then we have the B chain, then we have the C chain, and lastly we have a A chain. So this thing is called pre-proinsulin. So we have this thing also written here, which is the pre-proinsulin. This is pre-proinsulin togetherly is called. So it is available in humans as this form, and these are rolled in this. So when this entire pre-proinsulin is folded, this looks somewhat like this. You can see this, which consists of the leader sequence, which consists of the B chain, then we have the C chain, and then we have the A chain, and this A and B chain are linked by two disulfide linkages. Alright, so when this folding is done, so how will we get insulin? Because insulin is just these two. So I'll just rub it for you so that it's easy for understanding, right? Yeah, so I'll again select the black ink. There we go. So the insulin molecule is just this. You can see these only A and the B chain. So we just want the A and the B chain. Alright, and we don't want the C chain, right? We don't want the C chain and the leader sequence. But in humans, this is available in the form of this, which is folded, which would look something like this. But we want just this part, the A and the B chain. So what happens is, after this folding has occurred, translation happens. So after translation, what happens is, this leader sequence is removed. This particular leader sequence is removed, which cleaves the C chain as well. So you'll read here that, so the leader sequence is removed after translation. So after translation, so this is the folded thing, and after this translation will occur. So after this, what happens is, the leader sequence is removed. So this first thing is removed. With that, the C chain is also removed. So when leader sequence is removed, along with that, the C chain is also excised or removed. So the remaining part is only insulin that we have here, which is linked by disulfide bonds. So this is how humans retrieve insulin, alright, from the pre-proinsulin form. So this is the technique. So moving with that, so talking about the second application of genetic engineering, which is the human growth hormones. So we'll talk about the human growth hormones, how it helps us. So the logic behind the human growth hormones is same as insulin as well. So it is the logic behind this is to produce recombinant growth hormones. Alright, So there we go. So we'll start with, at about the same time that recombinant insulin was first being made in E. coli, other researchers were working on similar projects with the human growth hormones such as somatostatin and somatotropin. So human insulin are produced in E. coli. So I'll explain you on another video. In the coming video, I'll explain how the recombinant insulin is entirely made. So recombinant insulin is an application of genetic engineering, but I haven't explained the process of how recombinant insulin is made. So I'll make another video. In the next video, I'll be making about recombinant insulin. So talking about the human growth hormones, so the logic behind this is also same, which is the production of growth hormones such as somatostatin and somatotropin. So these two proteins act in conjunction to control growth processes in human body. So these two hormones are responsible for growth of an individual. Also, their malfunction leads to painful and disabling disorders such as acromegaly, which is uncontrolled bone growth, and dwarfism. So acromegaly is gigantism, you can say, or dwarfism is shortened height and all of that. So these two hormones are responsible for growth of an individual. And somatostatin, so talking about somatostatin, which was the first human protein which to be synthesized in E. coli. So somatostatin was the first hormone which was synthesized in E. coli. Alright, and being a very short protein, only 14 amino acid in length, it was ideally suited for artificial gene synthesis. So you can see here, so the entire thing you can see here. So this is the E. coli part, which consists of the lag promoter gene, we have the LAGZ gene, we have the artificial somatostatin gene, which is transformed in E. coli. So this is a basic bacterial DNA or bacterial plasmid, you can say. So this is a bacterial plasmid in which we have all of the things which are important. So lag promoter and LAGZ part I have already explained in my previous videos, so you can watch it. So talking about the lag promoter and LAGZ, and with the artificial somatostatin gene, which is transformed in E. coli. So from there, when it is transformed in E. coli, we get a beta galactosidase segment, which consists of the methyl region, alright, and it has a fusion protein as well. So from there we extract the somatostatin, and via cleavage with cyanogen bromide we get the somatostatin out, and this is how a recombinant somatostatin is produced. So this is another a great application in the human growth hormones, which is the somatostatin. So talking about the somatotropin, so this is a bit lengthy process as compared to the previous growth hormone. So in somatotropin presented a more difficult problem, which is this particular protein is bigger in size. So it is 191 amino acids in length, which is equivalent to 600 base pairs, an impossible prospect for DNA synthesis capabilities of the late 90s, 70s. So this is a bigger thing. So in fact, a combination of artificial gene synthesis and complementary DNA cloning was used to obtain somatotropin producing E. coli strain. So messenger RNA was obtained from pituitary, the gland that produces somatostatin in the human body, and cDNA library is prepared. So before moving into the diagram, I'll just explain you the context how it all started. So pituitary is the gland that produces somatostatin naturally in the human body. But in this case we want recombinant somatotropin in case the individual lacks this particular hormone. So the somatotropin cDNA contained a single site for restriction endonuclease He3, which therefore cuts the cDNA into two segments. So you can see this somatotropin cDNA fragment. So this is the preparation of somatotropin cDNA fragment. So the first process for somatotropin production of recombinant somatotropin is preparation of somatotropin cDNA fragment from the cDNA library. Alright, so we'll prepare a somatotropin cDNA library which contains a He3 restriction endonuclease restriction site. So it contains a He3 restriction endonuclease cleavage site on the somatotropin cDNA fragment, alright, which cuts the cDNA into two segments. So you can see here it is cutting the particular cDNA fragment into two fragments, into two unequal sizes. So you can see 0 to 24 base pairs or amino acids are in one and the rest of them in the another part. Alright, so first of all is the preparation of this particular fragment, then with the help of this particular restriction enzyme. So it contains a restriction cleavage site of He3 naturally, which cuts the fragment into two unequal parts. And what happens after that is the longer segment consisting of codons of 24 to 191 amino acids was retained for use in the construction of the recombinant plasmid. So this particular, the bigger part for production of somatotrophin was used for making of recombinant somatotrophin, and the smaller part was discarded. As you can see, this is discarded, but the longer part was retained in the production of recombinant somatotrophin. So this particular bigger part of somatotrophin was used, which was used by ligating a synthetic leader in front of the somatotrophin fragment. All right, so this is the bigger fragment. In front of that we have the synthetic leader which is ligated onto that, and then it is the overall in the entire thing is inserted into an expression vector. So in the entire thing is inserted into an expression vector. So this entire thing is inserted into an expression vector which contains the lac promoter. So we have the lac promoter here along with the expression vector. So when we use the entire vector, entire plasmid, and we transform it in E. coli, just as same we did in the insulin, we get the recombinant somatotrophin in this case. So this is very easy. So this is how somatotrophin is produced. So this is another great application of genetic engineering. So moving with that, so talking about the recombinant factor 8. So talking about the recombinant factor 8 and how it is very useful, which is the example of recombinant pharmaceutical produced in eukaryotic cells, is human factor 8. A protein plays a central role in blood clotting. So many of the individuals lack this particular factor, which results in blood clotting. So the production of recombinant factor 8 is very important for prevention of blood clots. So the commonest problem of hemophilia in humans results from inability to synthesize factor 8, leading to breakdown in the blood clotting pathway, and a well-known symptom known with this disease. So many of them suffer with hemophilia, which is blood clotting due to the lackage of recombinant factor 8. So the synthesis of factor 8 is very important, which we do in this case by synthesizing or by producing recombinant factor 8 to sort the problem of hemophilia. So until recently, the only way to treat hemophilia was by injection of purified factor 8 protein obtained from human blood provided by donors. So it was only possible if the donors would donate their blood, so that particular blood could be injected to patients who are dealing with this particular form of infection, which is hemophilia. So this particular infection was only treated by injecting the person's blood or the donor's blood, which contained the factor 8 protein. So the purification of factor 8 is a complex procedure and the treatment is expensive. Alright, so more critically, the purification is beset with difficulties, in particular in removing virus particles that may be present in the blood. So the purification of factor 8 that is present in donor's blood is also very expensive. It's not an easy process in removing all the viral particles and other contaminants that might be present. Also, hepatitis and acquired immune deficiency syndrome or AIDS can be or have passed on to hemophilias via factor 8 injections. So different sort of infections such as hepatitis and AIDS are transmitted via the factor 8 injections due to the presence of contaminants. So this particular filtration is very important for factor 8 for injecting into the person dealing with hemophilia. So factor 8 free from contaminant problems would be a significant achievement for biotechnology. So this is the goal that we need to achieve, is the filtration or removal of contaminants in the production of factor 8. So Talking about the another process, which is farming has been used for production of recombinant factor 8. So how can we produce recombinant factor 8? So we all talked about, until now we talked about factor 8. So now we'll talk about production of recombinant factor 8, which is achieved by the process of farming. So the complete human cDNA has been attached to the promoter for the whey acidic gene of pig, leading to synthesis of human factor 8 in pig mammary tissue and subsequent secretion of the protein in the milk. So this factor 8 produced in this way appears to be exactly the same as the native protein and is fully functional in the blood clotting assay. So this is in this case an animal. So I'll just repeat the entire thing, which is the complete human cDNA has been attached to the promoter of the whey acidic protein gene of a pig. So human cDNA is being used into this. So let's say this is a plasmid or a vector, you can say. In this, a human cDNA has been attached here with the promoter. So you can say you can take any promoter here for the whey acidic protein gene of the pig. So it has a gene of a pig also, right, leading to the synthesis of human factor 8. So this particular thing, so this production of this so the presence of human cDNA with the promoter and the whey acidic protein gene. So this is whey acidic protein gene of pig would result in formation of human factor 8. So this will lead to formation of human factor 8 in pig's mammary tissue and subsequent secretion of protein in the milk. Alright, so this is how we can produce human factor 8. So this production of human factor 8 is called the recombinant factor 8. Alright, so this would sort the problems of blood clotting eventually, and this is another way of forming factor 8, and also to get rid of issues such as filtration. But also we haven't actually got rid of filtration in this case as well, because contaminants might be present in this case too. But this is another way of producing another form of factor 8, human factor 8, via using acidic pig protein gene and production of factor 8. So let's just keep this video till here. Hope you enjoyed this. Thanks for watching.